Okay. People can hear me? Yes. No. Okay. Okay. Well, my name is Emma Rosi-Marshall, and it's really great to be here. It's been really great to get to know everyone here. It's been so much fun, such a great week. I've learned a lot, and now I'm going to tell you a little bit about what I do back in the U.S., and I'm going to focus on pharmaceuticals and personal care products, which is the title of my talk. I first want to just acknowledge what I'm going to talk about today. I may say I, but I should say we. This is a collective effort. This is research that's done in collaboration with the people listed here on my slide, many of whom are graduate students who deserve most of the credit for doing the hard work. I just get to talk about it. So I do want to acknowledge them first. I also put my email address on this slide, so if you want to contact me and have questions afterwards or comments about this research, please contact me. How do I make this go forward? Okay. I wanted to start by talking about urban ecology because this is something that the work that I do on pharmaceuticals and personal care products is centered in urban Baltimore and where I do research. But Fabio asked if I would talk a little bit about urban ecology in general, so I'm going to take a step back a little bit and talk about urban ecology and why we do urban ecology and some things that we can learn. And I'm going to talk about the approach we use in Baltimore as part of a long-term ecological research site. So many of you know this, but the world is becoming increasingly urbanized. So this graph here from Nancy Grimm et al., this was a paper that was published in Science. You can see that the rural population is going down and the urban population is rising. And in fact, we have now crossed that threshold, and now more people live in cities than live in rural areas. And so that's a fact of life. And so the Baltimore Ecosystem Study is a long-term ecological research site funded by the National Science Foundation. The funding started in 1997, so we've been conducting research for a long time. So similar to what Laura was talking about, instead of being on the floor of the ocean in the Gulf, we are in the city of Baltimore. So this, for those of you who don't know, this is a part of the Chesapeake Bay. This is the urban metropolitan Baltimore area right here. This research integrates biological, physical, and social sciences. So just to give you sort of a schematic of this, sort of the Tanslian ecosystem concept is that we connect the physical with the ecological. But what we do in urban ecology is we also think about the built environment, so the buildings, the roads, the infrastructure, and the social complex, the decisions that people make, the governments that we have, all of those things together interact and need to be studied when you want to study urban ecology. In Baltimore, the research that we do is to improve the understanding and application of the concept of sustainability to an urban system. We use multiple approaches. I'm going to talk about our watershed studies, but we study the internal dynamics. We do modeling and spatial analysis. We've reconstructed, for example, the land use history back to the 1850s in Baltimore. We study social science, so why people do the things that they do, why they make the decisions they do. We have economists. We study environmental justice, and we also do education and outreach. So these things all together are part of our research. Okay, what happened? Oh, where's my next slide? No slides? Oh, no? I'm going to keep talking. I know. Fabio says I can do it, so I will keep talking. Yes? Okay, so I'm going to just keep talking. So in Baltimore, we use the watershed approach, which was developed at Hubbard Brook by Gene Likens. So the idea at Hubbard Brook in a forested watershed was they took a whole watershed and they cut down the watershed to see what the importance of the trees were for nutrient export in that watershed. And so they took a whole watershed and cut down the trees and compared it to a reference. We use the same basic approach in Baltimore. I think I'm skipping a slide, but that's all right. Same basic approach in Baltimore where we want to understand, we designate the watersheds and we say the people have done the experiment for us. They have put a city there. You know, this watershed is very highly urbanized and this watershed is suburban, and then we have a reference so we can then, sorry, yeah, people can't see me. But so I will just move it like this. I'll sit here. So we use what's happening in the city as our experimental treatments for our watershed studies, and then we've studied them through time. And so I'm going to show you that in a minute as soon as I can. But basically, we have long-term records of nutrient export from suburban watersheds and from urban watersheds, and experimental treatments that we can then study. And so if I had a slide here, I would show you that in the forested catchments in Baltimore, basically we have very little nitrogen export. We care about nitrogen a lot in Baltimore because it drains into the Chesapeake Bay. The Chesapeake Bay is got a lot of nitrogen coming in, and it's basically promoting algal blooms in the Chesapeake Bay, and it's a real problem. Nitrogen is the major pollutant of concern there. Okay, let's see what we get. All right, excellent. So let me just step back. This is the slide I skipped because I didn't have it. One of the things, actually what I will do is I will go forward and then go back. So here's our watershed approach in Hubbard Brook. Here's where they cut the forest down, if you haven't been to Hubbard Brook, and here's where the forest was regrowing and they you know, left a strip of trees. And so we use the same approach in Baltimore. And there we go. So these are our streams. So here's a watershed that's forested. Here's a watershed that is suburban, and here's a very urban watershed. You can see the road over top. And you can see this is the forested watershed. You can see very little nitrogen coming out of this forested watershed. And here's the suburban watershed. And you see the nitrate concentrations up at 2 milligrams per liter, up sometimes to 4 milligrams per liter. This red one is an agricultural watershed, and I don't have the most urban watershed on this graph. But you can see that while urban watersheds are not necessarily as bad as agricultural systems, they're exporting a lot of nitrogen. This is a long-term record starting in 1997 and goes on into the future. What we've done, in addition to the experiments that already exist that people have done for us, is we also do other experiments by working with the city. So the city has this plan to become more sustainable. And one of their ideas behind becoming more sustainable is to retrofit the city so that it's not just a sanitary city where we don't have water pollution and waterborne pathogens. We actually have the city is more sustainable, so it's actually not such a large nitrogen source. And so we've done retrofitting of the city. So we put in green infrastructure that's supposed to remove the nitrogen. And so what we did in this watershed in Baltimore, you can see it's actually the streams are buried, they're underground, but they're there. So here are 2 sub-watersheds in this larger watershed called Watershed 263. These watersheds have a lot of empty, vacant row houses. So it's a pretty, we call it urban blight, where people have left the city and you have these abandoned houses and decaying infrastructure and lots of concrete. And so what they did in this study was they did a green retrofit in one sub-watershed and left the other watershed untreated. Right, so we put in, so here is a big backhoe putting in green infrastructure. So they take out the concrete and they put in soil and they put in plants that then will hopefully mitigate the nitrogen export from the watershed. And I don't have time to show you the long-term data and what was found, but basically we had a long-term record. Then they did this intervention where they retrofit the watershed, and then what we saw was a decline in nitrogen export from that watershed. But we coupled this to social surveys. So we we also asked the people in the neighborhoods what they think of their environment, and it turns out that when there are people putting backhoes and putting in green infrastructure, the satisfaction in the neighborhood improved. So people liked their neighborhoods more. And it's partly because the city was showing that they cared about their environment. They were making it more green space, and people were actually out walking more, which is healthier. So we had both environmental and social benefits. And this is sort of the idea behind urban ecology, that we want to both understand the ecological consequences and the human consequences as well. Okay, so I'm going to just go on. And so urban ecology, you'll maybe recognize this is where we are right now, in an urban area. I took that the other day. So urban ecology, the idea behind it is that we have long-term data to unravel the multi-factor complexity that happens in urban ecosystems. It's not just the biogeochemistry, but it's what the people do as well, and the decisions that they make. And that long-term data allow us to produce patterns or signals that can guide hypotheses and experiments, so that we have these data about the watershed export, and then we do an experiment where we do this green retrofit in one neighborhood versus another, and then we can do experiments and modeling to understand these complex systems. And then hopefully these data will help us understand how to become more sustainable. So Baltimore, we work directly with the city of Baltimore. They have a plan for developing more sustainability in the future, but they don't really know how to measure whether or not something actually influences the nitrogen export. And so we actually have the data to then do that research. Okay, so now I want to kind of shift gears a little bit. And here's that slide again of the nitrogen export. And here in this suburban watershed, so the nitrogen that's coming out of the agricultural watershed is from fertilizer. So you put fertilizer on the land and it comes out. Where is it coming from in a suburban watershed? Where is the nitrogen coming from? What's that? Lawns. Could be lawns, so partly it's lawn fertilizer, partly it's atmospheric deposition on the roads, but the big source is sewage. And the thing about Baltimore is that we have infrastructure, sewage infrastructure in the city, but it's leaking because it's old. So here, you don't have much infrastructure and you're putting new ones in. We put ours in 1908, and it's old and hasn't been repaired, and so it's starting to leak. And that's going to be a theme of this talk, the rest of my talk. So it comes from sewage. So here's some photographs of what it looks like when human sewage is in a river. This one is from here. The sewage that's coming from the city that we're in right now. So I took that picture on the bridge the other day. But here is the city of Baltimore. This area here where there is sewage in the river is from leaking infrastructure. There is no wastewater treatment plant in this watershed. It is all those row houses. Many of them are abandoned, boarded up row houses. You can see the crumbling infrastructure above ground. You can't see the crumbling infrastructure below ground. So there's a lot of sewage entering into the system from leaking infrastructure. It's not supposed to be there, but it is. This here is a city, this is in Nairobi, a river that goes right through the city of Nairobi. So direct inputs in some places and indirect inputs in cities where this infrastructure is decaying. So what else is in sewage besides nitrogen? Nitrogen is interesting. We all care about nitrogen. But I am quite interested in other things that are in sewage. And one of the things that the big class of compounds that are in sewage are pharmaceuticals and personal care products. So PPCPs. These are contaminants of emerging concern, because some people call them emerging contaminants. That's not really right. They've been around for a while. We just now think about them. So they're really contaminants that are now people are focused on. And so I'm going to talk about things like antibiotics, antihistamines, antidepressants, painkillers, anticonvulsants, antimicrobials. I'm not going to talk about hormones, but they're also fragrances and insect repellent, sunscreen. All of these things that we use in our daily lives enter aquatic ecosystems, have been measured in aquatic ecosystems. And I will hopefully convince you that they have ecological consequences. There are many pathways that pharmaceuticals get into waterways. You see, I come from the U.S., and I have in the middle of my diagram wastewater treatment plants, because in the U.S., most of our waste is routed through wastewater treatment plants or sewage systems. But wastewater treatment plants and sewage systems are not designed to remove pharmaceuticals. They are designed to remove organics and to not release ammonium, so to nitrify that ammonium, and so to release nitrate, which is less bad than ammonium in a river system. So these wastewater treatment plants, although sometimes they do remove pharmaceuticals, they are not designed to do so. So the sources are manufacturing facilities, which in the US and abroad typically wash down their equipment, and the pharmaceuticals just enter either a wastewater treatment plant or the river unchanged. So these can be major sources of pharmaceuticals. Then they get sold, and either we take them into our bodies or we use them on our bodies like soaps and fragrances, and they go down the drain. If we take them into our bodies, some pharmaceuticals are metabolized, meaning we break them down. So caffeine is metabolized by our system. So we don't urinate out much caffeine. We metabolize it in our systems. Well, why do you find caffeine in human sewage? Because we dump half of the coffee cups down the drain, right? Oh, I don't want to finish my coffee, it goes down the drain. So we have a lot of caffeine in our lives, so you see it in the sewage. It's just not coming out of our urine. But a lot of pharmaceuticals come out our urine or our feces. So they come through our systems. Because if you think about a pharmaceutical, if you take something in as a drug and you metabolize it in your stomach, it's not going to have its intended effect, right? It's supposed to travel through our bloodstream and go to our brains or do whatever. And so they tend not to be metabolized. A lot of other things that we use, fragrances and detergents, we don't ever go into our body. They're meant to go down the drain. We use them, and then they go down the drain, and so those enter the system that way. Again, wastewater treatment plants don't completely remove them, and of course here, 2 days ago I learned about the large amounts of direct inputs into aquatic ecosystems. And in the US we have direct inputs because our infrastructure is all leaking. So it's leaking, and that's directly adding sewage. And here you have sewage that's entering because you don't have the infrastructure or the wastewater treatment capacity. The other thing about wastewater treatment plants to remember is that wastewater treatment plants in the US and presumably here are designed to discharge liquid waste into a river system. The solid waste, if you can imagine what that is, so the non-liquid waste is then settled and then has to be disposed of somehow. And depending on the pharmaceutical compound, if it's a very hydrophobic compound, it will be associated with the solid waste. So if the solid waste is then land applied, which is what we do a lot in the US. So here's a big spreader of solid waste from a wastewater treatment plant. They spread it on the land for agricultural uses as a fertilizer. Then those pharmaceuticals get remobilized and then can enter aquatic ecosystems that way. So you've just kind of moved the problem from below the wastewater treatment plant to somewhere else. So when you think about wastewater treatment plants, it's important to remember that. The other thing to remember is that we use a lot, at least in the US, a lot of pharmaceuticals for raising animals, so cattle. I don't know if you do that here, but we do a lot in the US. And so a lot of our antibiotics are actually given to cows and chickens. And their waste does not go to a wastewater treatment plant, but rather goes directly into aquatic ecosystems. So there are many, many sources of these drugs to rivers and lakes. So this is that river again in Baltimore that I work on, and you can see, I like to talk about the trash you can see and the trash you can't see. So you can see here's an orange juice container, plates, there's hypodermic needles, there's lots of trash in this river, but what you can't see are the high concentrations of drugs in the water as well. There was a paper that was published in 2002 that's been cited like, I don't know, 2,000 times at that point, of all of the compounds that were measured. So they measured something like 90 compounds in surface waters in the US. These references here, if you want to write them down, are ones that are found where people studied it in Brazil. I looked just last night about Brazilian, so these are a couple places where they have measured pharmaceuticals in Brazil. And not very many, I wasn't able to find many. But I assure you there are pharmaceuticals in the water because there are people here. People use drugs, drugs get in the water. I also want to show you, this is a very small stream that's quite beautiful and forested that drains through the Cary Institute property in New York where I work. And even though it looks like there's no people, no urban, no trash, you can't see any trash, but there are pharmaceuticals in this river as well. So they're found in many places. So what I do as an ecologist is I try and understand what the consequences are of these drugs. There are many, many papers showing that drugs are occurring in waterways. That's not new. What is new that I'm going to talk to you about is what do they do. So it's like saying, you know, as a biogeochemist, well, I measured nitrate in the water. Fine, okay. What does it do? What does it matter? Does it matter that there's nitrate in the water? Does it do anything to aquatic ecosystems? And that's what I'm trying to address, is what do all these drugs do to aquatic ecosystems? And I hypothesize that they do things like affect the mortality and growth of organisms, behavior, community structure, they move through food webs, they might affect ecosystem function, they might affect biogeochemical transformations, and they might interact with other stressors. And this is a big area of research potential, or that my colleague and I have written a paper about. And I'm going to just show you some examples of research that I've done in my laboratory where we try and tackle this very, very big issue. Okay, so the way that I approach this is I think that we need to use what I call multiple scales of inquiry to do this research. Toxicologists tend to work in single species, and they dose them with individual compounds, and they look and see what concentrations of compound will kill them. That's fine, important work. Fine. I'm an ecologist. I don't do that. I do something a little different. I like to go and do things like incubate sediments in the laboratory, or ideally go out in the field and do exposure. So I'm going to tell you a little bit about small-scale field exposures. Oh, that's a picture that I don't know where that came from. But artificial streams, I have. That's not a picture of an artificial stream, so I don't know what that's from. It's strange. And I'm going to talk about work that I've done downstream of wastewater treatment plants, and then finally using environmental gradients, and then doing large-scale controlled additions of experiments. I'm not going to talk about that. I've only done one. There's plenty of drugs out there. I don't need to add them. And here would be a great place to do research on pharmaceuticals, because I'm sure they're here. And what I'm going to talk about is how we investigate the effects of these on aquatic ecosystems at multiple spatial scales. So first I want to tell you about an experiment that I do that's really similar to what Laura was talking about when she did the oil exposure down in the deep sea. So instead of exposing things to oil, I'm going to expose biofilms in rivers to pharmaceuticals. And so what we do is we take a cup that's about this big, and we add agar and pharmaceuticals to the agar. Then those pharmaceuticals diffuse slowly out across a porous substrate, and biofilms grow on that substrate. We can then put those things out in the field, and then the biofilms grow, and you can look at how the pharmaceuticals influence the structure of the communities and the activity of these communities. And so we measure things like the chlorophyll A, so the algal pigments. We measure the gross primary production. We're starting to measure the composition of the algal communities by looking at diatom species, and we've looked at heterotrophic respiration and microbial communities that I'm going to tell you about in a minute. This method is published it was published last year, and we have another methodological paper on using contaminant exposure substrates in general that's in review right now. Okay, so we did this experiment. We were interested in just common drugs. People asked me why I chose these drugs, and I said because people use them. I didn't really have a hypothesis about, except for antibiotics. Antibiotics should suppress activity of bacteria, but I didn't really know what caffeine, and this is antihistamine used for allergy medicine. Tagamet is an antihistamine used for heartburn, so for stomach upset. And then I used an anti-diabetic and another antihistamine. And then we added them into these pharmaceutical diffusing substrates. And I'm just going to cut to the chase here and tell you what the data showed. And that is that basically the responses varied by compound. So here's primary production. These are the different compounds that we exposed the biofilms to. We did this in a relatively pristine stream in New York just to see, well, what do drugs do to these activities? And what you can see here is this one here. This is diphenhydramine, and you can see we had about a 98% reduction in gross primary production. We exposed them to this diphenhydramine, which is Benadryl, which I believe is very, very commonly taken here as well. So this, you know, if you have an allergy attack, or for sleep medicine as well. We also saw a significant reduction, in respiration. here you can see by many of these compounds affected the microbial activity, so reduced the activity of these microbial communities. The other thing that we're able to do with these contaminant exposing substrates is we can then take a little piece of that stuff, that biofilm, and we can actually do pyrosequencing, or Illumina sequencing is what we're doing now, to look at the microbial communities. So we can look at the bacterial or algal communities on those substrates and see if there's a difference in terms of what grew there. And what you can see from this graph here is that what we got, we did this in 3 different streams in this experiment, 3 different places in Indiana, in New York, and in Maryland. These bacterial communities on the control substrates were very different from each other. Then when we exposed them to this diphenhydramine, the communities were homogenized. So the bacterial communities became much more similar to each other. And basically it's because we had an increase in a Pseudomonas species and declines in 2 flavobacteriums. What's interesting about flavobacterium is they're known to degrade algal carbon. So remember I told you that this diphenhydramine suppressed algal activity, so it's not surprising that we saw a decrease in the bacteria that like to utilize algal carbon. But that's, again, that research is published. Now I'm going to talk about a different scale that we use, which is artificial stream experiments. So we recently built an artificial stream facility at the Cary Institute. We have 20 artificial streams. If you want to do experiments in artificial streams, please come. We like to have people come to my lab and do this kind of research. This is what they look like. They're about 4 meters in total length. You can put whatever substrate in that you want in the bottom, and it's a paddle wheel that moves the water around and recirculates the stream. It increases the scale of your experiment, the duration of your experiment, ecological complexity. You can put bacteria, algae, invertebrates. You can measure ecosystem function. You can measure biogeochemical transformations in these streams and expose them, of course, to drugs. And that's what I'm going to talk about now. I'm going to tell you about a couple different experiments that we did. I don't have time to talk about all the ones that we're doing right now, but I just wanted to tell you about a few. One is triclosan. How many people, at least in the room, have heard of triclosan, this compound here? Has anyone heard of it? 1. Okay, because I told you about it. That doesn't count. Okay, so triclosan is the active ingredient in antibacterial soaps. So how many people have seen an antibacterial soap or used it? So it is in your tissues, and it's probably in your urine. It is similar, it's a hydrophobic compound, and so it accumulates in our fat tissues. So and it can be passed through breast milk, for example. So it is very commonly found. It's been found all the way into dolphins, so it will biomagnify in food webs. It's similar in structure to PCBs or DDT. I don't mean to say it has the same ecological effects as those compounds, but it's similar in structure. In fact, it's an endocrine disruptor. It affects the androgen system. So it affects testosterone and your responses to testosterone. So it's a little bit different than an estrogen mimic. So we wanted to know in this experiment what it would do to bacterial communities. So we did an experiment where we added triclosan. Again, I don't know what that is, but it's showing up in my slides. So this is a paper that was published in Environmental Science and Technology this last year. We added triclosan in realistic concentrations to streams that had active algal and bacterial communities in them. So we grew these communities in the streams, then we added triclosan and we saw what happened. What you can see here is this is over 34 days. What you see is not a big decline in the numbers of bacteria. This is an antibacterial compound. We didn't kill the bacteria. So why is that? Because, on the next slide I'm going to show you, we had big resistance to triclosan. And it happened actually over only 7 days we got, that was our first measurement. We see triclosan-resistant bacteria. And for those of you who don't know much about bacteria, bacteria have this really neat trick where they can pass genetic material across species. So if it's attached to the plasmid, there's antibacterial resistance. If it's attached to the plasmid, they can join and share that genetic material across species. So you can get resistance spreading through bacterial communities very, very quickly. So it's rapid evolution. So it happened in only 7 days, and you can see big, fast resistance to this compound. We also saw a big change in the community structure of these communities. So for those of you here that were on the field trip yesterday, cyanobacteria bloomed in our artificial streams when we added triclosan. So we had a big increase in cyanobacteria. So we looked at the microbial community structure. Again, that paper is published in ESNT. Some other recent findings that we have over this last summer and work that was done by a former graduate student of mine, we have seen that antihistamines affect invertebrates and their population dynamics. We have recently demonstrated that amphetamines, so amphetamine is both a licit and an illicit drug, so it's used in speed. If you know what that's a street drug in the U.S., and it significantly reduced primary production, gross primary production. Another antihistamine significantly reduced GPP, and then we have found recently that antidepressants, so Prozac, I don't know if you know of that drug, but Prozac is very commonly used in the U.S., significantly suppressed GPP, community respiration, and increased chironomid emergence rates. So we put nets over these streams, and we can measure how many bugs actually emerge, and we got a big increase in a number of emerging chironomids. They emerge much quicker than in the controls. Why it's happening, we have no idea, but it definitely seems to have an effect on aquatic insects. Okay, just reminding you, now I'm going to go up to a higher scale, and we're going to go out in the field. And these are some experiments that we've done in the field. So this is in Chicago. Again, this was published in that Drury et al. Paper on triclosan. So this is taking advantage of a natural environmental point source. So here is a Chicago metropolitan area. This is the second largest wastewater treatment plant in the world. We are zoomed very far out. So you can see all these circles. Those are all the aeration ponds. It's the second largest wastewater treatment plant in the world. It treats 250 million gallons per day. The first largest wastewater treatment plant in the world is on the south side of Chicago. So we have the two biggest in the world in Chicago. So we sampled above the wastewater treatment plant and below. And then we went to a very small wastewater treatment plant and sampled above and below. And we looked at the microbial communities. And what, we measured the sediment triclosan concentrations. So we went out and took sediments, measured the concentrations of triclosan, and then at the same time measured the microbial community's resistance to triclosan. And what you can see here is that out in the environment, when there's triclosan present in the sediment, you get triclosan-resistant bacteria. This is the first evidence of triclosan-resistant bacteria in the environment that we know of published. The other thing I want to point out is that we also went to a wooded stream where there was no triclosan, we're pretty sure, and you see that there is triclosan-resistant bacteria there. And it's because bacteria, the way that they are triclosan-resistant, there's a natural method that they use to become triclosan-resistant, and that is with these efflux pumps to get rid of large carbon molecules out of their tissues, and so or out of their cell, and so there's natural resistance to triclosan, so that's why it can develop so quickly. So some bacteria have this. I do want to point out that the highest concentrations of triclosan in the sediment were not found below the wastewater treatment plant, but actually were found above the wastewater treatment plant. And that is because wastewater treatment plants do a pretty good job of removing triclosan. But above the wastewater treatment plant in Chicago, there are combined sewer overflows. And I don't know if you're familiar with that term, but what we do in the U.S., or what we have done in the past, is we mix, when it rains, so that not all that rainwater goes to the wastewater treatment plant, we mix the rain and the sewage and we release it directly to a river. And where above the wastewater treatment plant, there's a lot of combined sewer overflow, and so the concentrations of triclosan are actually higher above the wastewater treatment plant than below, which is sort of an interesting observation. Okay, now I want to talk about using the streams in Baltimore as a natural environmental gradient to study these pharmaceuticals. And so in Baltimore, we have a network of buried streams. So here, this is where the stream, this is the headwaters of the stream, if you will, because it's all been buried underground under the city of Baltimore. Comes out right here. It's got lots of sewage. In the drainage of Baltimore, we have 500 storm miles and 900 sanitary sewer miles. Those were put in about 1908. We don't know where they are. They're not mapped, and they're leaking. And so many of them are in a state of decay in the U.S. So raw and untreated sewage enter streams, and so it provides a nice gradient. Just to demonstrate to you that there are pharmaceuticals, this is work that was done both with an LC mass spec and a GC mass spec by one of my graduate students. And you can see here the concentrations of caffeine. So 1 microgram per liter of caffeine, that's a lot of caffeine in the river. Our phosphorus concentrations are about 3 or 4 micrograms per liter. And then we find things like amphetamine, diphenhydramine, a whole suite of compounds. This is ecstasy, which is a popular street drug. How am I doing on time? I have no idea. 3 hours. Great. Excellent. So this is also, what's that? Don't worry. Okay, good. So here's another illustration of the gradient. There's another way you could take a grab sample to measure pharmaceuticals, but you can also use an organic, it's called a polar organic contaminant sampler, integrated sampler or something. So it's similar sort of to what Laura presented, where you deploy something and basically the pharmaceuticals diffuse across a membrane into this sampler, and then you can bring it back to the lab and then extract them. And so what you can see from this is I show you the nanograms per POSIS, so per sampler, so it doesn't give you a per liter concentration. But along this urban gradient, you can see that caffeine is really high in our very urban streams, and acetaminophen, which is a painkiller, and antibiotic as well. We also have, as I told you, amphetamine. There's a big drug problem in Baltimore, and we can see it in the streams. So we see morphine, which is a breakdown product of heroin. So when you inject morphine into your heroin into your system, your body rapidly converts it to morphine, which is why you get a very fast buzz off of that drug. And what's quite interesting, and there is a paper published from Manaus in Brazil, a couple papers that I found on sewage epidemiology. So people, if you ask them, do you take drugs, they say no. But if you look in the sewage, you find cocaine and crystal meth in the sewage, and so it's this new emerging field of sewage epidemiology. So we use the sewage to figure out what's going on with the people. And we actually just wrote a review on the ecological effects of these kind of illicit drugs, so I published that this summer if you're interested in these kind of drugs in the environment. Again, just to show you that we have a nice strong gradient of pharmaceuticals along this urban gradient. So here's amphetamine and diphenhydramine, just as an example. We also find antibiotics in these streams. So those, I didn't show you antibiotics in that last slide. Antibiotics are found both in the urban streams, but we also find them, here's this beautiful forested, we call it our reference site. Our reference site it's out of the city of Baltimore. There's not supposed to be much urban activity, but there are some houses with septic tanks. And in those, when there's no septic fields, we don't find hardly any antibiotics. So these are fluoroquinolone antibiotics and ciprofloxacin, which is a very commonly used drug. I'm guessing it's taken here a lot. It's a powerful antibiotic. When you have septic fields, you also find it in the environment. And this is cipro-resistant. So we find antibiotic-resistant bacteria in the sediments in a stream that looks like this. No city around. It looks like a nice place. We do see the consequences of these antibiotics. And then this is the rest of our urban streams, just to show you that we have antibiotics and we have antibiotic-resistant bacteria. Now I'm going to go back to this contaminant exposure substrate so you can deploy them along a gradient, and that gives you the opportunity to look for the ecological response of communities that you're pretty sure have been exposed, right? So by doing it along a gradient, I can say, well, I know that these microbial communities are not exposed very often to drugs, and these are exposed very often to drugs. And so I can then deploy these and look and see how the communities respond to a known drug. So I can say, well, I know in my little cups here, I have an antibiotic, and then I have a control. And how does a community that's very urban respond to those drugs versus a community that's doesn't see the drugs very often at all? Does that make sense? So it's sort of an approach along the gradient. So we deployed these contaminant-exposing substrates in 4 streams along this urban-rural gradient. We added caffeine, cipro, which is this antibiotic. You've all heard of ciprofloxacin, maybe? Okay, we'll have to look at your drugs because I'm sure it's used here, but it may be called something else. Diphenhydramine, cimetidine is an antihistamine. And so we grew heterotrophic biofilms in the streams, and then we measured the respiration on these sponges as an indicator of the functional response to these drugs, and then we did Illumina sequencing to look at the microbial responses. And this is a paper that we're writing right now. So I'm very much interested in your feedback because I'm writing the paper right now. So what we saw along this gradient, so here's our urban-rural gradient. I need to orient you to these graphs. So this is the response ratio of the respiration. So because the control in all of these streams would be different, we don't want to just show control treatment. We want to actually look at the control relative, the treatment relative to the control. So we do a response ratio. So if it's a 1, that means that there's no difference between the treatment and the control. So the bacterial communities didn't care that the drug was there. They had the same respiration when exposed to the drug as when not exposed to the drug. And so I want to just highlight this ciprofloxacin. So this is this powerful antibiotic. And what you can see in the urban stream is when these bacterial communities are exposed to this antibiotic in the stream, there's no effect. They have the same respiration rates than if they weren't exposed. Now, one can argue that the control is also being exposed, right, because of the overlying water, right? So it's a little funny because you've got both the stuff that's coming up through the pharmaceutical diffusing substrate and the pharmaceutical that's in the stream water. But I put a lot of Cipro in my agar, so I'm pretty sure they're getting exposed mainly, they're getting a lot more in my Cipro exposed than in the stream water. And so we see no effect. Versus the rural stream, you can see a significant suppression relative to the control. So in the rural stream, you get a big suppression in the rate of respiration. And we see that the same, not as much of a response of these, but certainly these are significant declines. But then when you look at the microbial community response, we find some really interesting things. So these are the different drugs, and this is a change in the diversity of the microbial community. And what you can see is that you get, across the board, these drugs decline, suppress the diversity of the microbial communities. And you can see that Cipro does a very strong suppression. These sites are arranged along the urban gradient, so rural to urban. And you can see that Cipro causes a big decline in the diversity. But in some of the sites, not so much. There wasn't a big decline in the diversity. And it depends on the drug, it depends on the site. So the responses are going to be different. And then when you actually look and see which species or which groups of bacteria, and I apologize, this is a very busy graph, but when you do Illumina sequencing, you get tons of data back. So we have lots and lots of data on the whole microbial community using 16S rRNA is what we sequence. And what you can see here, so this is the response ratio of the community of bacteria. So if it was the same as the control, it would be at a 1. And if it's higher than the control, then that means that on the Cipro, there were more of that type of bacteria than the control. And what you can see here is that, like Pedobacter has gone up in response to Cipro, whereas Pseudomonas has gone down. Regardless of which site, Cipro seems to cause an increase in Pedobacter and a decrease in Pseudomonas. But at some of these sites, you can see that you see a significant decline in some groups, but no decline in the most urban sites. So there are some indication that these, like this one, for example, right here, looks like it's probably resistant in the urban stream and not resistant in the rural streams. We're still trying to make sense of these data. The other thing that's quite interesting about this is in laboratory studies, we know that Pedobacter and Basia are known to be resistant to fluoroquinolones. So this is the antibiotic, it's a Cipro, it's a fluoroquinolone, and it's known that these specific bacteria in laboratory conditions are resistant. And in the field, we found that in all the streams, they showed that they're resistant. So it's a really nice complementary study to what is known in the lab. Out in the field, we can demonstrate this same response. And this, again, some taxa decrease at the less urban sites, but not at the urban sites, suggesting that there may be resistance. And this is those same data shown on an NMDS plot. So for those of you who aren't familiar with this, it kind of, it takes all the data, the entire assemblage of bacteria, and it puts it as a dot on the map here on this this NMDS plot, and dots that are closer to each other are more similar in terms of their microbial community. And what you can see here, these are the 4 study sites, so these are the sites. They're all different. They all have different microbial communities. But when we expose them to Cipro, those communities all start looking a lot more like each other. Again, this homogenization of these bacterial communities. But this is the most urban site, and it changed the most. It had, and again, the exposure to Cipro had no effect on the function, but the community changed the most in the most urban stream. In contrast, in the least urban stream, it had the greatest suppression of respiration, but the community didn't change as dramatically. And we are proposing this idea. We're calling it the pollution priming hypothesis, that pollution that we know is occurring in this urban stream, it doesn't happen all the time, the concentrations are low, but what it does is it promotes some resistant bacteria that are kind of hiding out there in the urban stream, and when you expose them to the drugs, they're able to then grow and develop communities very quickly and maintain that functional resiliency that we see in this urban stream. This is what we're putting forward. We don't know if it's really right, but okay, so I just want to finish with this slide, which is sort of a thought map that I've walked you through today. There's increasing human populations in the world, and of course in urban areas in particular. I didn't tell you this, but the sales of pharmaceuticals, and this is just prescription drugs. It doesn't include illicit drugs, and it doesn't include antibacterial soaps, and it doesn't include fragrances and all those other things, sunscreens, coffee, all that other stuff. But it's $1 trillion in the U.S. No, $1 trillion globally in 2014. In the developing world, we have aging infrastructure and wastewater treatment plants that are not designed to remove drugs, so we get drugs in that way. In the developing world, that either they lack infrastructure or the infrastructure is getting built, but again, they're using the same technology that we use for wastewater treatment plants that are not designed to remove these drugs. There are some methods that we could talk about good ways to remove them. And so what we end up with is increasing concentration of drugs in surface waters worldwide. It's certainly happening because all these different pathways are going to increase the concentrations. But what we don't really know are what the ecological effects are. And I hope I convinced you today that there are effects of drugs. Most of the things that I do, I try and do them at environmentally relevant concentrations. So our artificial stream experiments, we did that at concentrations we observe in the field. Environmentally relevant changes a lot, though, when you go from one place to another. My colleague that works on drugs in rivers, he's a chemist, and he's working in the Ganges. And he said he went to the Ganges to take samples in India, and he brought them back. And he totally underestimated the concentrations of drugs. He has to go back and take samples because his concentrations were way too high. And so the concentrations in India below manufacturing facilities, for example, can be in the milligram per liter concentrations. So there's can be really high concentrations of these drugs. And I think, and I have demonstrated there's effects on bacteria, algae, invertebrates, ecosystem function, nutrient cycling, probably behavior. The list goes on and on. Really interesting research. And so I say that this is a talk that's to be continued because there's a lot more to be done. And with that, I'm happy to take questions. So thank you. Questions? I don't need to do anything, right?
And now I can talk. Okay. This is not the first time I watched this presentation, not exactly this presentation, but it was really nice and to watch it again, and I wouldn't be bored to watch it every month or something like that. It was really nice. But the question I have is that, have you ever investigated how the community recovers as you move downstream?
No, but it would be a good thing to do. So we, in that paper I told you about trickle, that was a student named Brad Drury, and he published 2 papers below the wastewater treatment plants that he looked at the microbial communities. One is the one on triclosan, and the other one is in applied environmental microbiology, so in AEM, which is another journal. I can send you that reference if you're interested. And in that, we just looked at the microbial communities downstream of the wastewater treatment plant. So we went above, and I think, I'm not sure if we went to more than one site below the wastewater treatment plant. But what we saw in that study was, again, what we think happens with sewage is we got a homogenization of the microbial community. So we went to 2 rivers above the wastewater treatment plants. They were very, very different bacterial communities. And below the wastewater treatment plants, they were basically indistinguishable from each other. So we think that sewage, and this will be something I would be very interested to do, to compare, you know, for example, in the rivers here. Like if we take samples where we know there's human sewage here in Brazil and compare them to microbial communities that we've measured in the rivers in Chicago, are they similar? Because human sewage is human sewage, and that actually we have a homogenization globally of microbial communities, and I predict that that would be the case. Because even though, you know, we eat different things, whatever, pretty much our sewage looks a lot alike in terms of the labile carbon. But I think it would be very exciting to do, you know, how does the microbial community change along a gradient in terms of recovery. Now, one thing that we are doing right now is we're doing the microbial community along that urban gradient at the Baltimore Ecosystem Study. So even though we've been doing research there for, I don't know, 17 years and studying nitrogen, everything about nitrogen biogeochemistry that you could want to know about nitrogen, we have never opened the black box of the microbial community and looked inside and seen what's in there, who are the microbes. And so we've done, we currently have a paper that we're writing doing metagenomics, so not just the 16S rRNA, but we're sequencing all the sequences of the bacterial community and looking at how the communities differ. We're still waiting for the data to come back from the array because it's a huge amount of data. And then we're also, my postdoc is looking at the diatom communities and the bacterial communities along this urban gradient to see if we can see some interesting differences in the microbial communities. But certainly I think there's a lot to be done. The one thing I can say is that unlike macroinvertebrates or fish, which don't like urban areas, right? So you typically see a decline in the diversity of these kind of things in urban streams. We think we actually see an increase in the diversity of bacterial communities or in urban streams because you have all these crazy weird compounds going into the urban streams. You have labile sewage, you have oil and gas and PAHs and all these things. And so what you end up with is a very diverse assemblage of bacterial species.
Bacteria?
Yeah, yeah.
Okay.
Yeah. So we think, but we're working on it. So.
Is everybody listening to me? Okay. First, thank you very much. I'm very happy to have the opportunity to listen to you. It was very, very interesting. And I have a couple of questions. First, I was thinking from the beginning, like trying to relate what you do to what we do. And even though these compounds are dissolved in the water, do you expect them for, like when you have a watershed and you dam a river, should them like be trapped behind this dam, or because they are dissolved they would just flow freely? I mean, does the dam change, I mean, accumulate it and then diminish the amount of it downstream somehow?
Okay, first of all, let me answer that question and maybe I didn't say this earlier in the talk, and I should have said this, which is that pharmaceuticals and personal care products is a huge category. We're talking about 3,000 at the minimum number of compounds, all with very different chemical structures. So they range in whether or not they're going to be dissolved in the water column versus whether or not they're going to be absorbed to sediments. Their range in terms of their persistence. So triclosan that I mentioned is very persistent. If you do sediment cores, there was a really nice study where they did sediment cores in New York Harbor. The patent was approved in 1963. They find it in the sediment cores in 1963. So it's definitely persisting. And so they range. And, caffeine is probably it's probably not persistent at all because we metabolize it and so do bacteria. It's actually a pretty easy compound to use for a bacteria, but other compounds are not. So the range is going to be huge. So if you have sewage entering a system, some of it's going to be in the dissolved phase, some of it's going to be associated with particulates, some of it that's in the dissolved phase may be attached to particulates once it gets into the water. So if you put a dam there, you surely will see the ones that are associated with particulates will fall out and be in the sediments. I think that's absolutely the case. It's going to depend on the compound. And if you're interested, I can kind of give you here are the list of things that I would look for in the sediments versus here are the things that I wouldn't bother looking for, because of the structure of the compounds and their use patterns. We use a term with these compounds called pseudo-persistence. So we say caffeine is pseudo-persistent, meaning like pseudo, P-S-E-U-D-O, like fake persistent, pseudo-persistent, pseudo or whatever. And so because of the fact they don't persist in the environment, but they look like they persist because no one stops drinking coffee. We keep putting it in. We keep every day more and more comes in, so it's pseudo-persistent. So they're always there. Does that make sense? Yeah. So just to clarify, there's many, many structures, many different potential fates.
Yeah, and then maybe related to this, like when you have these experiments and you have certain amount of chemical there, and then you have your bacterial metabolism. In the end of the experiment, the amount of this chemical is the same there. I mean, the bacteria, usually they don't break this compounds, or do they? I mean, are them going somewhere, or they're remaining in the ambient?
Okay, so it depends on the experiment. In the pharmaceutical diffusing substrates, it's basically passive diffusion, and those pharmaceuticals are going out. And in this new paper that's in review, they follow Fickian diffusion kinetics. They go like this. In the first couple of days, a lot diffuses out, and then we strive for this, having our experiment during that linear, where it approaches linear, so the diffusion rate's about the same, and so that we kind of hope for our experiment is in that time frame. Certainly, the bacteria that we measured in that last experiment where I told you we looked at antibiotics, some of those are known to degrade that antibiotic, so they may be living on that substrate because they're getting fed by our diffusing substrates. In our artificial stream experiments, we have a great opportunity. We added 1 microgram per liter at the beginning, and then all we did was add water to account for evaporation. So we didn't add any more water, and it was a contained system. And after 3 weeks, we saw something like 70% of the original concentration of the amphetamine. So amphetamine seemed to be relatively persistent in that environment. But we could, I think we are going to, my student is going to take the algae from that experiment and try and extract the amphetamines and see if we can find, do they get degraded or are they just sorbed to the organic substrates? So you have the opportunity to study these fates in an artificial stream experiment for sure. So we have done a little bit of that. Yeah.
I'm wondering if it just travels through rivers to the ocean and then it accumulates all the time, or if it's being degraded and disappearing somehow. It's nice.
Yeah. So there's one study where people measure the spiraling length, so how far these compounds travel. There's just one of those. We have ideas that we would like to do that. I don't think they probably I think it depends on the compound. Some compounds may just travel unchanged all the way to the sea, and some might get degraded. I think it really depends. Some are degraded by light. There's one compound that's very interesting that's called the zombie compound, because it degrades in light and then in the dark it reforms. So we thought that it was degrading, but then when it gets dark it reforms its structure. So it's really strange. Yeah, varies a lot.
Thank you.
All right, can everyone hear me? Yeah. I was wondering a little more about your experimental design. Um, Particularly, I had kind of two questions about this. I saw in your pictures there you have these trays, and on the trays you have the little. The fact that they're so close together doesn't cause any problems.
Yeah. So, okay, they are next to each other, and we put them so that they're like this, and the river flows over them like that. So yes, the drugs will diffuse out, but we take advantage of the unidirectional flow of a river to make them go that way downstream. And then once they diffuse into, so it's a very small amount of drug diffusing into a very large volume of water. And so we really, we've calculated the diffusion, again, in this paper. We know how much goes out, and it's this teeny fraction every day, you know, like the amount that would cross-contaminate controls. So it's, you have to think of it as it bathes the biofilm in a high concentration, and then it goes into a river, and it's so dilute that we don't think it has an effect. The fact that we see big differences between our control and treatment suggests we don't see carryover effects.
Okay.
Yeah.
Could you maybe also, where is the biofilm is it growing just on that sponge there in the center? And so with that, you kind of have to cut that's just growing right on, nothing grows outside of the foam on, for example, onto the plastic that's surrounding it?
Well, it would, but what our the sponge, or we use another thing that's like a glass thing to grow diatoms on. They grow better on the there.
Right.
That's like our sample, right? So we know that there surely is biofilms that grows on the outside, but we don't care about that because it wasn't exposed to the drugs. We just use that little thing as our sample unit, and we take, for measuring production and respiration, that whole thing goes into a light dark bottle, and we measure, you know, typical ways that you measure GPP and R, and then we just take a real small subsample of that to get the microbial community.
And then just one more question on a kind of a bigger scale. What are some things we can do to obviously is take less antihistamines, take less antibiotics, use less antimicrobial soaps, and etc. But is there any other non-obvious ways that we can help this situation?
Yeah. The one big thing in the U.S. is we need to repair our infrastructure. We have spent, I mean, you are all now putting infrastructure here in the city, and it's like you invest all this money and time and effort to route the waste to a wastewater treatment plant, and we did that, and then we just like out of sight, out of mind, we don't think about it anymore. In the U.S., the American Society for Civil Engineers has given our sewage infrastructure a D+ as a grade. I don't even know where the plus comes from, but a failing grade. We are not maintaining it at all. And so that is one thing that we could really do. I mean, we want to invest, we want to protect our systems. We should invest in the systems that we have built. And of course, here, we should build systems. You know, we should put the waste into a wastewater treatment plant. As I told you with the triclosan, wastewater treatment plants, though not designed to remove drugs, they can remove some drugs, especially something that's really hydrophobic. It'll absorb to the sediment, the biosolids, and then of course you have to dispose of the biosolids knowing that they're contaminated, so you should do something like incinerate them. But that is something that we can do. The other thing is, so we should build wastewater treatment plants and build infrastructure. The other thing is we should fix our wastewater treatment plants and invest in new technologies to actually remove pharmaceuticals. So they're not currently designed to do that, but they can do that. So it's been shown that wetlands can remediate some of these drugs, can remove some, and the wetland plants can take them up, and then you can harvest those wetland plants and incinerate them properly. So there are ways, especially the Europeans are very far ahead of us in working on methods to remove pharmaceuticals from wastewater. So it's possible, but usually wastewater is the last thing. If people aren't getting sick, people are not investing, the government's not investing money in it. That's just all there is to it. So,
okay.
Yeah.
Everyone hear me? Yeah? Can you hear me? I don't know. Can you hear me? Why? No, it's okay. First, I'd like to say a huge thank you for both Laura and Emma, and it was really great to have both you here, and very nice talks. And Emma, I have I think actually I have 3,000 comments, but I'm doing just three. First, I'd like to have now 19 years old, just to be in the second level of the undergraduation and to be here. And I would put in my mind, so first I have to improve my English very fast because I want to talk with this teacher. I want to work with this teacher. And the second, I would write a letter if I would be a student and say something like this: Emma, I'm 19 years old. I was in your 19 years old. I was in your talk there, here actually, because I would writing from here, and I supposed to finish my undergraduation in 2 years or some. And do you think I would have some way to visit you at Cary as a student or a trainee or to work in the lab? That's my first question. The second question is uh, regarding a kind of conceptual question. It's really to put in just one question because my mind is completely like a centrifuge, 5,000 speed per second, many things in my mind. And when we showed us the resistance and some bacteria questions, I try to think aquatic systems as a metaphor or as a explanation for any system like a human system. For example, when we took drugs and drugs and drugs, bacteria inside us to become resistance. And in the system, seems to me, it's a similar process, maybe in different scales, maybe if you understand the synergic in the system, it's much more complex probably than in the human bodies because we took drugs for specific pathways in our body, in our human systems, I would say. But in aquatic systems, we don't know exactly where the drugs are be strongest trouble or response. You know what I'm saying? So this is really, really important because we have, as scientists, as a student, to be very clear about this in order to explain to the people that make decision. For example, when we work with bacteria, okay, for biology, for undergraduation, for ecology program, there is no problem to talk about bacteria or even about virus. But if you talk about bacteria with some engineering, they say immediately some disease or some applied, okay, using bacteria to convert something from something to another product, but it's not the case. Bacteria and virus and actually cyanobacteria as well, and many other populations and communities, they are pieces of the game in the ecosystem perspective. So we need this conceptual improvement. It's not, you know, in order to get more partners to save the waters? And the third question would be exactly the Zach said about the treatment plants, and you mentioned. So I have two questions, in fact, this conceptual, and if I would go to a age tunnel, go back, and now I have 19 years old, and I'm gonna write a letter for you.
Okay. 3 questions and 3,000 over dinner later. The first question is, I mentioned that I had lots of students to thank. I am at an institute in Millbrook, New York, and I have this artificial stream facility, and I have a wonderful lab, and I have a million ideas, and I have not enough time to do them. So I very much welcome students. So I have a student from Nebraska, had a student from Australia, a student who contacted me and said, can I come and work in your lab? And I said, sure. And she decided to measure emergence from these experiments. So there are formal opportunities, and then there are informal opportunities to work in my lab. I very much enjoy interacting with students. And one thing that I think that is really exciting about this research is that it's daunting, but it's exciting. There are 3,000 compounds to choose from, and all of their interactions, which is however many orders of magnitude more, 3,000 with the exclamation in mathematics, right? So I could be busy until I am 100 doing experiments. And so my student that is working on Prozac, she's a student from Australia, and I said, well, what drugs do you want to work on? And she said, I'm really interested in antidepressants. And I said, great, let's work on those. This other student said, I'm really interested in illicit drugs. Great. And then I wrote a paper about illicit drugs because I realized there wasn't anything. So there's so much research that needs to be done, and there's really great opportunities. So I welcome people to come to my lab. It would be great to have more people. And the artificial stream facility is a great place to do experiments. Question 2. So I intentionally have a picture here with many different drugs. And I think when I speak to the general public, I give a different talk. And I talk about cocktails of drugs. And that we know in ourselves, we know, one, we should finish our antibiotics, right? Because if we don't finish the antibiotics, right, they say to take all of your antibiotics, because if you don't, you will promote antibiotic resistance in your body. In the streams, they're getting antibiotics and then there's none, and then they're getting some and then there's none. And so promoting antibiotic resistance in streams is similar to ourselves. The other thing that we know, because I don't know if it's on the ads for drugs in Brazil, but in the US, when there's an ad for a new drug, at the end of they say, it's going to make you happy and your life will be better if you take the drug, right? But then at the end of the ad, they say, don't take this drug if you're taking blah, 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 all these other drugs, right? Because it will kill you if you take them together. Our streams are always getting a cocktail of drugs. So I call it a cocktail. It's a mix of drugs. They're getting many different ones in lots of different combinations, and we have no idea what the effects are. So I told you only a little bit about, we're starting with one drug at a time. My postdoc right now just did an experiment. I only told you a little bit about her diphenhydramine, but she also did a combination of diphenhydramine and salt, because road salt is a big pollutant in urban streams. So we put road salt when we have ice, and then it washes into streams. And so we know that salt can influence bacterial communities and algal communities. Well, what happens if you have salt and a drug, and you mix those together? How does the community respond? And I'm really interested in doing the entire cocktail of drugs, putting all of them together, and then putting them with really high concentrations of nutrients, or with really labile carbon, because all of those things are what happens in the environment. It's a cocktail. It's a recipe for, we don't really understand what it does. We don't know. So I think it's a really exciting area of research. It's going to be really hard to do, but I agree with you that I actually think that talking to the public about drugs in the water is a great way to talk to people about nitrogen in the water. So when you tell people there's cocaine in your river or there's birth control pills in your river, they get concerned. If you tell them there's nitrogen in their river, they don't know what nitrogen is. They don't know that they have it. They don't know that they need it. They don't know that they excrete it. But they know that cocaine is bad. You know, they know that birth control pills do something, right? And so I think it's a great opportunity, actually, to connect the public with their waste, if you will, and their rivers. So I look at it as an opportunity. So it's a great way to engage the general public. And your third question, I have forgotten. Oh, the treatment. Yes. So there are many, 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 many papers in the scientific literature on developing new treatments, ozonation, UV. Some of them, like UV, can actually make the drugs worse because UV can break some drugs down into more potent and more toxic breakdown products. So that's a problem. Some things like artificial wetlands can work for some drugs, but not for others. So there's a really rich literature. I don't keep up with it because it's just something in addition. But there are certainly opportunities. And I think that it also provides a nice opportunity to say, well, traditional wastewater treatment plants, don't remove them. So maybe when we're talking here, we need to think about some options for a wastewater treatment plant of the future that could do something about these drugs. You know, if we build wetlands and attach them to the wastewater treatment plant, could that then mitigate this as well, and think far into the future? But that's going to be a challenge, but I think that there's certainly a lot of research being done to figure out what waste treatments can remove pharmaceuticals. There aren't that many in place yet, but.
Sewage waste treatment plants. But when we have just for, we don't take out the nutrients. So nowadays, we must to rewritten our sentence. The third or the last part of the sewage treatment must take out nutrients and pharmaceuticals. So good don't have for nutrients. Otherwise, we should add the new step. And in this case, it's playing with this scenario that we don't have, and say to the people, okay, take out the organic matter, anaerobic process, but also nutrients and other very dangerous compounds to the water that actually it's much more not visible than the other ones. If you give some water to a polluted water for someone, if completely clean, they can take it. But if it's a lot of, the smell is not good and the color, so no, I don't want this water because blah blah blah. So the government or the responsible to treat the water, they considering that part and give the visible nice water. But we have to use that visible clean water. In fact, it's much worse.
Yes.
Because there are many these kind of things on it.
Yeah. Again, I think there's an opportunity. Zach asked, about people taking less drugs, and people should take less drugs. I think we take a lot more, and we certainly use antibacterials and all these things that we probably don't need. But we are never going to stop taking drugs. If people are sick, they should take drugs to get better. I don't think that we should not take antibiotics. If you're sick, I take antibiotics. I mean, we should take drugs. So that means if we're always going to be a source and drugs are always going to be a part of human sewage, then when we treat our human sewage, we should treat this other part of the sewage, the drugs. Just because they're in low concentrations does not mean they won't have an effect. And that's sort of the, right now people say, oh, but they're in low concentrations. But a low concentration of a drug is what we design drugs to be, low concentrations and have an effect. So it doesn't make sense to say it's a low concentration, it won't have an effect. Yeah. Are there other questions?
No, I have one. We're talking about how the humans compounds, or how the compounds that human has been used affect the environment. But my question is, do you know if there is literature or any data that prove these compounds in the environment affect the human again? Like, is anyone die or something happen with the population?
I don't know of examples of humans. I do know examples of birds. There was an example of vultures in India where it wasn't through the aquatic. It was basically overuse of pharmaceuticals, and then they were in the cows, and the cows all died, and the vultures. So there was that example. There haven't been. There's no evidence that it affects people from drinking water. Maybe because people aren't really investigating it. I don't really know. The other thing, in the U.S., people always ask me about the quality of their drinking water, and they're very concerned. Like, they care more about the drinking water that they have than about the ecological condition. So I focus on the ecological effects because I'm an ecologist and that is what I do. But a lot of times people, when I talk to the general public, they say, well, what about my water? Is it clean? And a lot of times these people live in New York City, and they have one of the best drinking water supplies in the world. They don't really have pharmaceuticals in their water. But I would also argue that the amount, this is for the U.S. I'm not talking about Brazil now because I don't know here. But in the U.S., our drinking water is very, very clean. It is very good, it is very highly regulated, and indeed people have measured pharmaceuticals in drinking water, and it's very low detection rates. So people are also trying to figure out methods on the drinking water side to remove pharmaceuticals before the drinking water, but they're very low concentrations. But what I often say to people is, so you're concerned what's in your drinking water that comes out of the tap, but you don't know what's in your food. You don't know what kind of pesticides are in the hamburger that you eat, or even, you buy makeup. It's not regulated, stuff to put on your skin. You don't know what those compounds are. You don't know what the fragrances are. So there's a lot of things in our lives. We live dirty lives. Like, all these things that we use, we have no idea, but we want our drinking water to be pure H2O, right? Which wouldn't be good to drink. But, right, there's this idea that this water should be perfect, but everything else, I have no idea what's in my, what I'm taking, what kind of drugs I'm taking, what kind of things, pesticides I'm exposed to. And so I try and educate people about that. Now that's may not be the case here. It may be that the drinking water has issues and these pharmaceuticals might be in the drinking water. I don't know. I have not seen that being measured here. The few citations that I found from Brazil were not measuring it in the drinking water. So, that could be an issue here. But I don't know of any cases where it would have an effect, or where they've shown an effect on people. Again, the thing about the drinking water is it might be a nanogram per liter, at least in the U.S., it's nanogram per liter concentrations. And you know that if you drink a nanogram per liter concentration of caffeine, it's not going to do anything to you, right? We know we drink milligram per liter concentrations in our coffee. Now, if it's cancer treatment drugs or other, maybe you don't want to have birth control in your water. Some people might want to, but maybe you don't. And so we don't really know. Yeah.
Yep.
So has there been any research to see, like, are there any, you've been, I think, Rafael asked about spatial effects, right? And downstream, are there any farms that you've looked at to see if well, for example, the cattle are drinking the water from the stream, if it bioaccumulates in them?
I have not done that, but there have been some studies in agricultural systems looking at concentrations of pharmaceuticals in the water below animal, like cattle and things like that. various agriculture, and with land application. There's been some studies on that. So what, as I mentioned in the slide where I talked about that paper that was published in 2002, you know, we'd all be so happy to have our dissertation cited 2,000 times 10 years after it was published, right? There's something, the EPA, the US EPA, curates a list of all the papers that have been done on pharmaceuticals in the environment, and they're currently up to 15, 16,000 citations. So there are many, many, many studies of pharmaceuticals in the water. No doubt, all over the world, people have measured it. The ecological consequences or consequences for animals drinking the water, people, harder thing to study and not as well studied.
We are finishing here. Thank you, Emma, again, and all of Laura for this wonderful talk.